Log on, tune in, find out. Another good idea from Cambridge.
And he'll be talking about the critical path, biomarker development and streamlining clinical trials.
Thank you, Stephanie. So you noticed that my the announcement had TBA for the title. So my attitude is I want to get as much information as I can. And so while Val and Andy were talking this morning, I was composing my talk in the back and including the title. So this is hot off the presses. My financial disclosures, I have a company that designs adaptive trials for cooperative groups and pharmaceutical companies and medical device companies. This is you all seen these kinds of things before. This is the overall success of phase 2 and 3 in the pharmaceutical industry for the last 7 years. Notice the horrible performance of oncology. 34% of phase 3 trials are successful, and that's largely because of the lousy development in the early phases, the critical path, which of course is...
That's what success means. Yeah.
Success means you run a trial, it has a goal to show statistical significance, and it doesn't. Now, some subsets of these therapeutic areas are worse than others. Alzheimer's, if you're familiar with the process, is pretty bad. I attended an Alzheimer's Association workshop a month or so ago where they presented the last 13 phase 2 trials that led to phase 3 trials. All 13 phase 3 trials failed. And if you look at the phase 2 trials that led to them, you start snickering. There are all kinds of things going wrong. The development programs, by the way, were very dear. GSK developed Avandia, rosiglitazone, the diabetes drug, much in the news in the recent years. They developed it for Alzheimer's disease. $500 million spent on the development of the drug in Alzheimer's disease. And if you look at the phase 2 data that led to phase 3, you'd be appalled. A very common problem is subset analysis. And it befalls the big companies as well as the small companies, although the small companies have few statisticians to clue them in, and so the small companies tend to be to fall more prone to the problem. Why phase 3 failures? Half the time, these are completely subjective on the basis of my observations in the industry. Half the time it's because the drug itself is ineffective and they get to phase 3. Sometimes it's not a dumb thing to run a phase 3 trial. Alzheimer's, for example. If you ran a phase 3 trial that was successful, the nugget at the end of the day is billions of dollars. So it's like playing the lottery. And if you had a 10% chance to win the lottery, you might do it. So it's not necessarily irrational, but maybe you could build a better program that would be more informative and adaptive in the course of phase 3 and getting out and if it's not going to be successful. So half the time it's an effective drug and half the time it's an effective drug, just a lousy development strategy. And these are my reasons, the silly Of course, many times it's a number of these together. So you could have the wrong endpoint. You could have a single-arm trial in oncology historically, although it's getting better. Historically has been based on single-arm trials, looking at tumor response as the endpoint, which not always translates into a clinical benefit. Preserving jobs. So I have this, when I speak with pharmaceutical companies, when I'm talking to the top echelon, they want to know if a drug is a dud, so they can get off and do other things. When you talk to the team, the pharmaceutical team that's running the trial, or developing the phase 2 part, wants to go into phase 3, and you tell them, I just spoke to God, and she told me whether your drug is effective or not. Do you want to know the answer? And I don't actually say this, but think of my saying this. This is what they would be thinking. If the answer is yes, then that's good. If the answer is no, what am I going to do next year? I'm going to have to get a job at another company, or this one's going to move me to Siberia or whatever. So the motives in the pharmaceutical industry are not necessarily consistent with the overall company's good utility. My slides are available, by the way, if you want to, or if they're not on the website, you can get them from me. So I presented this at the Alzheimer's Association to kind of address this silly subsetting. So Ulysses, you remember, had himself tied to the mast of the boat when they're approaching the rocks with the sirens, and he wanted to hear the sirens, but he knew that they were susceptible to him, coming under their spell and mesmerized and crashing into the rocks. And so what he did is he had his men tie him to the mast, and for his men to put wax in their ears so they wouldn't be susceptible to the sounds of the sirens. So here you see Ulysses. See his eyes. You see he's bound by his men to the mast, and they're holding him. They told him to attack him if he breaks loose. And wait, I went too fast here. Yeah. So these are the statisticians. They're driving the design, etc., and they of course have wax in their ears. And this is the protocol. We used to notice the statistician is making sure that Ulysses obeys the protocol, that he stays according to the So this is sirens and subsets. I must say it was a bigger hit when I presented it to the people. So this is Janet Woodcock, who was and is the director of the Center for Drugs at the FDA, lamenting, as I've been doing and many people do, the sorry state of phase 3, and suggested that improved utilization of adaptive and Bayesian methods could help resolve the low success rate. She too and others, Sue Jane and others, have been involved in the FDA's Critical Path Initiative, and this is an opportunities report. This is where I got the title from. FDA uncovered a consensus that the two most important areas for improving medical product development are biomarker development and streamlining clinical trials. So she and others at the FDA have been pushing both of these initiatives. This is a paper she wrote with two of her colleagues in the New England Journal, highlighting interestingly, a so-called groundbreaking trial in breast cancer to predict drug responsiveness based on the presence or absence of genetic and biological markers, I-SPY 2. And I designed I-SPY 2. I'm a co-PI of, the co-PI of I-SPY 2, and I want to tell you about that. And Kyle Wathen is going to tell you more. The FDA, in part because of the critical path, has an initiative on regulatory science, and they funded 4 grants recently with the NIH. So there's an FDA NIH initiative for improving drug regulation. So this is a science piece that describes our grant proposal, successive grant proposal. Roger Lewis indicating our current approach to trials is horribly inefficient. We need to do something better. Lewis and Berry, along with emergency medical person Bill Barzan, University of Minnesota. This is a cooperative group called the NET, the N-E-T-T, the Neurological Emergency Treatment Trials Network, that run trials in stroke, in epilepticus, status epilepticus. I gotta take a drink. Spinal cord injury. And so we're designing adaptive trials for them, but we're also, as it indicates here, we're addressing the barriers. Where do we run into problems? And I can tell you there are many, having to do with people not understanding, people saying, well, we're adaptive. You know, what can you add to us? Are there problems with patients not understanding or not participating? There, I think there are no barriers. Physicians, there are. Investigators in learning the process. Many investigators come in incredibly enthusiastic. Sometimes, occasionally, as in the case of Alzheimer's, the investigators are too enthusiastic. I mean, they think you can create a good drug from a bad drug. And so, you know, there are no miracles here. Current use of Bayesian adaptive designs at MD Anderson, we've had over 300 trials. Jack Lee and I had a paper where we chronicled the first 5 years of the new millennium, and something like 200 trials taking a Bayesian approach, and most of those were adaptive Bayesian. Device companies, as Andy indicated, there's a guidance from the Center for Devices at the Center for Devices and Radiological Health at the FDA, saying how to do Bayesian things, and it morphed from using historical data back in 1997 when the initial impetus occurred. They thought that they could use historical information. Turns out to be very difficult, and we can talk about that, but there are problems associated with using historical data having to do with selection bias. And it morphed from that into adaptive Bayesian designs. Drug companies, as many of you know, essentially all drug companies are getting on the bandwagon, if it is a bandwagon. Andy and I in one conference, I think independently, came up with the hula hoop comparison. 1957, there were 15 million hula hoops sold. In 1958, there were 15. And so I hope the adaptive hype doesn't go in that direction. I don't think it will. And these are, one is the guidance from the Center for Devices, the other the guidance that you're all familiar with from CDER and CBER. So Andy mentioned this trial, and just to say a bit more about it, Frank Giles, when I first got to MD Anderson a little after 2000, Frank said, I want to run a trial. I have this new drug, troxacitabine, for AML, acute myeloid leukemia, and I want to compare it to standard therapy. But I want to do it in pairs. So standard therapy is idarubicin, Ara-C. I want to take troxacitabine and pair it with idarubicin. For a second arm, I want to pair it with Ara-C, and I want to compare it to the standard therapy, idarubicin, Ara-C. And he said I wanted 25 patients in each of the arms. And I said, well, why don't we look at the data? And if it turns out that one of the experimental arms is doing poorly relative to the control and relative to the other one, we'll downweight it as we go. And if it turns out to be doing sufficiently badly, we'll stop accrual to that arm. And if two arms drop, then we'll stop the trial. So we did adaptive randomization to learn while effectively treating patients in the trial, we thought. We assigned with higher probability, as Andy has mentioned, using a modification of this Thompson procedure. By the way, notice the 1933. I've looked carefully at the paper, and I'm interested in what Andy thinks, because I guess he has too. I looked carefully at the paper to see if he said the word or said words consistent with randomization. Because, you know, randomized clinical trials were in the 1940s with Bradford Hill. So does Thompson get precedence, or should he be mentioned in the same breath? So he talks about a proportion being assigned rather than randomization. I still don't know the answer. I don't know whether to give him credit. In the trial, TI, one of the experimental arms, was dropped after the 24th patient came in. So we didn't have CRs on everybody at the time, but we had enough information, you know, the endpoint at the time, but we had enough information to drop TI. After the 34th patient, the trial stopped because TA was zeroed out. And these were the data. TI had 0 for 5. If you ask the question, could TI still be better? Do we make a mistake here? Could TI be better than IA? Historically, IA was about 56%. The answer is sure. We could have made a mistake. The probability that it's better is very small. And if it is better, it's certainly not much better. So I don't think we made a mistake, but there's a caveat to that, which I'm going to come to. If you compare with the Korn-Friedland thing that Andy mentioned, said if you do two-arm trial and you compare how many patients, what the power is, so you build a trial with adaptive randomization versus equal randomization, and you power the two the same, then you're going to have more patients in the adaptive randomization arm, of course, and so you'll be treating more patients with the inferior arm. So they focused on treating with the inferior arm, which in my editorial, one of the things I pointed out was that's not the appropriate comparison. A more appropriate comparison is to say, of these 75 patients that were putatively going to be part of the trial, how did they benefit from the trial? So in the adaptive randomization, remember there were 18 in IA and 11 in TA and 5 in TI. These were the CRs. The other 41 patients had to be treated somehow, weren't part of the trial, but presumably got IA because the trial stopped on the basis of futility for troxacitabine. And so if this is just using a maximum likelihood estimate as to how many CRs there would have been, there'd have been about 48% CRs in these 75 patients. If you compare to balanced randomization, there would have been 28%. Now, if you have a drug that improves the performance of AML patients by 20% CR, that would be huge. So we both found out that the drug was a dud and treated patients better than had we done otherwise. I want to point out that this Thompson procedure is not optimal, regardless of the C that you put in that power. That honor goes to Bandit Strategies, and to my amazement, Bandits have been applied not only in clinical trials, but are now being applied by search engines. Google has about 20 people who do Bandit Strategies. They invited me out to give a talk a few months ago. So why are they interested? So you know when you go to Google, there's an ad, and the ad has various characteristics. Sometimes it's the same company that's doing the ad, but it's different as to what they show. They might show in one a watch, you know, they're selling watches. They might show in another one a young lady with a plunging neckline. I mean, they show all kinds of things. They get paid by hit. So when you click on that thing, they get paid. They want to ensure, however, the longer term. They'd like more hits in the longer term. So they'd like to learn as well as make money. And those two things, those conflicting desiderata, go into the Bandit strategy. You know, if you've got 100 observations and you can learn initially, how do you configure, how do you choose what to show in order to maximize the total income over the whole 100 hits? Of course, they have 100 showings. Of course, they have many more than 100. So Steve Scott is at Google, and if you're interested in some Bandit stuff, this goes back to 1972 when I was 12 years old. I want to talk about the external impact of the trial. There are occasions in data monitoring committees where a data monitoring committee historically will stop a trial and the credibility goes down because they stopped the trial. And and somebody else runs another trial doing the same thing. Here's a case in point. The company had nothing to do with the company. I worked with Frank Giles. It had nothing to do with the company. The company saw these data and didn't believe them. In part, I suppose, I don't know, in part because small sample sizes. You know, this 0 for 5. Frank sent the paper to the journal Blood, and the editor said, you can't do anything with 5 patients. I said to Frank, send him a note and say, if he gets this disease, we have a treatment for him. But he didn't do that. He sent it to JCO and JCO, Journal for Clinical Oncology, and they said, clearly the drug is a dud, but the design minimized the exposure of patients to inferior therapy, and so we're going to publish your paper. Maybe the company thought like the first editor, and they built a phase 3 trial, a so-called phase 2-3 trial, and the DSMB found that it has never been published. This is all I know, this press release. They verified what we told them had they listened. So there's a bit of a contradiction here. In a way, we built an efficient trial, one that would stop as soon as possible, minimize the number of patients exposed, but maybe we increase the number of patients exposed to the inferior therapy by not having a solid thing that solidified the question. Some areas of application of Bayesian adaptive drug trials. Migraine, of course, or something like status epilepticus, where you get almost immediate data on the endpoint, the registration endpoint, like 2 hours pain-free in the case of migraine, is ideal for the adaptive approach. Oncology and many of these others, the endpoint is longer in coming, and so it's more difficult to run an adaptive design, but it's more important, the biomarker stuff, it's more important to get information as to how the patient is doing over time and actually model, something we've done really poorly in medicine generally, model the patient's outcomes over time, even though they're not the fundamental primary endpoint, they're potentially informative for the final endpoint. Simulations are usually required, as Andy so nicely pointed out. Prospective design is essential. Longitudinal modeling, this idea of looking at the patient over time. Many scenarios, accrual rate matters. This is my very subjective assessment of the, in order, of the uses of Bayesian adaptive designs in the medical research generally. Of course, stopping early or late, you know, extending accrual. You get to the end of the trial and you look at the data and you don't know the answer. Not the answer you want to know, but you don't know whether you've got a drug. P equals 0.06 or something like that. You go to the FDA and they say, well, we believe in your drug, but your trial was underpowered, go do another trial. Wouldn't it be nice to have built that in as the possibility of extending it to get an answer? Or in one case I know of, the company did what the FDA said, ran another trial, they came up with P equals 0.07, and they went back to the FDA, and the FDA said, you know, we believe in your drug, but your trial was underpowered, go do another trial. So they suggested, well, maybe we could combine the two studies. Didn't work. They had to do another trial bigger than both of the previous trials, and of course, eventually got approved, but should have been adaptive. Dose finding, the Aston trial is one, but many others. Seamless phases, and these are not exclusive. That is, there are trials, you know, you could be dose finding and then seamless, and even population finding. I put this one in. The bottom one, remember I said these are in order of use. I've never done it. I've never seen an adaptive design that's built on choosing an endpoint. The reason I put it in is because in the last two Alzheimer's meetings I've been to, Christine Sampaio from the EMA, Portugal, insists, she's incredibly enthusiastic about adaptive designs, but she insists that this is the main thing that you should be looking at in adaptive design. Where is your drug effective? Is it based on ADAS-Cog? Is it based on MRI? Is it based on some other measure? Why do they do it? Smaller trials, more accurate conclusions, address more questions. What is the dose? What is the schedule? What is the subpopulation that benefits, if any? What about concomitant therapies? And it's not a major focus in many studies, can focus on better treatment of patients in the trials. Here's two recent examples using predictive probabilities and longitudinal modeling. This is an example that's probably the hardest to do an adaptive design. It's elderly breast cancer, adjuvant breast cancer, Patients have wonderful prognoses relative. In fact, there are elderly breast cancer studies that show that the mortality in those studies is better than actuarial mortality.
What do you mean by better?
Pardon me?
It's a better.
Pardon me?
What do you mean by what? Better mortality.
By which?
By better mortality.
Speak Russian, I understand better.
Better mortality.
Better mortality. That is, they live longer if they have breast cancer than if they don't have breast cancer.
Okay.
The endpoint here was a recurrence of the disease, and it was a study that was difficult to run. So I was working with the NCI and trying to set it up. We had funding for it through the NIH, the Institutes of Aging, and we said we wanted to, this was comparing capecitabine, which is an oral agent, with 2 chemotherapies, doctor's choice, CMF and AC, if you know the lingo, or even if you don't know the lingo. And we said 600 patients. And the NCI said, no, you can't do it. You need at least 1,800 patients. And we said, we can't do 1,800 patients. It's going to take us 15 years to get 1,800 patients, and by then nobody's going to care. And they didn't budge. So I finally said, okay, so here's what we'll do. We'll build a trial. We'll start looking after the 600 patient comes onto the trial, and we'll ask the question, will we know the answer? Is capecitabine non-inferior to the standard chemotherapy or not? Will we know the answer with an additional follow-up period? And if so, then based on Bayesian predictive probabilities. And if so, we'll stop accrual. And we'll make the decisions, reasonably liberal, nothing like the usual O'Brien-Fleming boundaries, for example. And we are less subject to type 1 error, although we accommodated the control of type 1 error. We're less subject to it because in O'Brien-Fleming, if you cross a boundary, you yell eureka, and we win. Here, if we cross a boundary, we're still subject to the underlying parameters because we follow for an additional period of time. So the type 1 error rates are less, there's less penalty to pay. And we built the trial. We got the 600 patients. We looked at the data. We said, we're going to know the answer with probability at least 80%. And so we're going to stop accrual. We followed. We did for 2 years. At the time, 600 patients, we had about 50 events. So a very small number of events, but enough to be predictive. And 2 years later, we had a compelling answer. I mean, these predictions, people complain about predictions that, things can happen. So far, in the many predictions that I've done, they've been smack on. Call it luck if you like. But remember I said when I spoke to God there? And so, it was sufficiently compelling that we got to publish in the New England Journal positive results. These interim analyses were not the standard type in which the trial results are announced when the boundary is crossed. Rather, the decision to discontinue enrollment was based on a prediction that the future follow-up was likely to give a meaningful answer. And I should have pointed out that word Bayesian occurs in the abstract. I'll come back to that. This is a study of an ablation catheter that was used to get approval from the Center for Devices at the FDA for this ablation catheter for treating paroxysmal atrial fibrillation. And the design was a prospective, multi-center, randomized, unblinded, Bayesian designed Study conducted at 19 hospitals of 167 patients. The company approached us when the trial was really in its early stages, saying we're accruing so slowly and we've heard that you guys can do magic. And so we worked with them to change it into a Bayesian design. It was originally 230 patients. We got positive results, I'll indicate that, but when we submitted to JAMA, JAMA said, well and good, but we want you to do confidence intervals and tests of hypotheses, and you can't use, we've never used the word Bayesian in the abstract, you can't use the word Bayesian in the abstract. And I was, of course, livid, and I wrote back and said, you are a medical journal. You should be committed to the science of medicine that says that you must follow the protocol. The protocol was Bayesian, completely Bayesian. You must give the Bayesian conclusions, and then you can do whatever you want, saying that the hypothesis generating or the also rans. And I shamed them into it. So the word Bayesian, I think for the first time in the Journal of the American Medical Association, appears in the abstract. And this is from methods. Design with perspective and Bayesian protocol defined primary statistical analysis, and primary endpoints was Bayesian. The error rate was controlled at 2.5%, verified by simulations. Maximum sample size was 230. Pre-planned interim analyses to occur after 150, 175, and 200. The first time we wrote it, it was to occur after 100 patients. But I have to admit that it took so long to get agreement with the FDA as to this change from the previous design to this one, and the fact that there were already some patients that had been accrued, how to handle those, because there was some information available from those patients. So all of those things took a while, and so by the time we got to get the design actually in, we were up to 150 patients. And again, the same idea. If we, based on a three-piece exponential model, where the early outcomes, there are many more early outcomes than late outcomes, and so there's more information about what's happening there. But we did longitudinal modeling for the patients that didn't have complete data. If they got through the first month, for example, they were likely to be successes at 9 months. This is a report in the, from the Institute of Medicine last year about the NCI cooperative groups, and the things that you may have read about. The report said we have too many cooperative groups, we would compress them. But there were some other things that they addressed. For example, the sorry state of the design in oncology trials in cooperative groups. We need better phase 2 trial designs to more accurately assess which patients benefit, to guide decisions about whether to move to phase 3. This could lead to faster, more accurate conclusions. So one of the things that they proposed, actually I have to admit I proposed, because I was part of this committee, was something like this. So you're interested in a factorial, you're interested in whether an agent 1 or agent 2 is better, or the combination is better. So you start out doing frequent interim analyses, possibly dropping arms, possibly doing adaptive randomization as you're going. You might drop agent 1 and continue with just, a partial factorial. You might drop the combo and continue with just agent 2 in a confirmatory phase. An example very much like this in diabetes from the critical path that Sujane Wang knows well is type 2 diabetes, seamless phase 2-3 design, dose finding, and dose finding using adaptive approach based on information to get information about what is the maximal utility dose, what's the minimally effective dose, both of those things. Sample size between 200 and 1500, active comparator, placebo. The primary endpoint, and one of the things I added, primary, in view of Val's talk, primary endpoint was a clinical utility index. So the registration endpoint is HbA1c. But the company is also worried about, and patients are worried about, weight gain. So weight gain is not part of the regulatory aspect, but they couldn't sell a drug if it was associated with substantial weight gain. So that was another one of the components of the utility index. There were 4 total, HbA1c, weight gain or loss, and 2 safety endpoints, heart rate and blood pressure. So the phase 2 was based on, we're looking at 7 doses. The idea was to pick 1 or 2 doses to go into phase 3 in the confirmatory phase, seamlessly, never stopping accrual. In fact, the same DSMB is looking at the data over time and then is just informed that you now have only 2 doses in the trial. They can still drop one of the doses if they want to, but if they drop after the cut point, when there's a switch to phase 3, then you go forward with only one dose. And so that they were concerned about that because they knew when the thing was coming, when the possible analysis was coming. And indeed, it picked 2 doses at the earliest opportunity. Sample size for the continuation was based on predictive power using the available phase 2 data. Of course, available phase 2 data was not complete for all of the patients because the endpoint was at 1 year. And oh, by the way, longitudinal modeling was critical because the choice of the doses was to occur before the first patient got to 1 year. Adaptive transitions, and this is just a little bit of the clinical utility index, and it was really quite amazing setting this up. I was not optimistic. The company was talking about, well, you know, we worry about weight gain, the FDA and we worry about safety, but we can't really put those together in any kind of way. And I said, well, let's think about it. Let's see if we can. And so this is the utility. This is the utility associated with HbA1c in comparison with the active control. And so if the active control is sufficiently better, then you have no utility. Similarly for, I'll try this thing. Similarly for weight loss, if there's a sufficient weight gain, then you don't have any utility. If you have sufficient heart rate increase relative to placebo or blood pressure increase, you don't have any utility. And these things are multiplicative, so it's treating them as being independent. And we showed various possibilities, and they loved it. I mean, it was one of the easiest things as part of the design. And the FDA accepted it for the entire development, including into the phase 3. The only thing the FDA worried about was, you know, where you draw the line as to heart rate or blood pressure. And so that was their only comments, and we changed accordingly. How am I doing time-wise, Stephanie? I'm over?
I think you're almost over time now.
Okay, I'm going to go to iSpy 2 and just tell you a bit about it. So this is a Wall Street Journal article with a nice graph. I say nice graph. I made the graph, so. But it says, uses genetic profiles to highlight biomarker differences among patients and to match drugs to patients with biomarkers that predict a benefit. The idea is to have, this is the standard approach in the top with huge phase 3. The idea is to use many arms, comparison with control, identify which arms benefit which subsets of patients, if any, and to end up with small, focused phase 3 trials, focused on the subpopulation that benefits. This is just to show you that other people have tried their luck at, this is the American Association for Cancer Research, at making a similar kind of graph. There's lots of hype associated with this trial. I didn't have anything to do with this one, although it seems to copy some aspects of the Wall Street Journal article. So when the first patient came into the trial, we had a press conference, and I presented this cartoon. This is the standard approach where there's a population of patients and an experimental arm, or maybe randomization, better of course, but still it's a homogeneous set of patients. I don't know how many papers you've read in cancer that start out with, cancer is a heterogeneous disease, and then the rest of the paper treats it as homogeneous. We focus on the heterogeneous aspects. What happens if you treat it as homogeneous? 34% success rate in phase 3. So here's what we do. We start with a number of experimental arms. The main focus here is this population of patients. I'm sorry, I did all kinds of bad things. This population of patients, which is now heterogeneous. And so we would, arm 2 may graduate into a small focused phase 3 trial. Arm 3 may drop for failure. It doesn't benefit any subset of the disease. Arm 5 may graduate into a separate subset. We add arms as we go, so it's a screening process. And we're doing adaptive randomization within the individual biomarker signatures. You could do this with, say, a factorial design. So the last 4 arms here, standard of care, and C and D and C plus D, this is a factorial design on top of the study. And the goal is 85% success rate. Entire focus is on phase 3. What is the success in phase 3? And if it's sufficiently high, then you graduate into a biomarker signature. And the biomarker signatures are sort of complicated. There are 10 biomarker signatures that we've identified that are subsets, that are combinations of subsets of these that I mentioned. Laura Esserman is the PI. The context is neoadjuvant breast cancer. We currently have 3 drugs moving on, experimental drugs moving on to 4. This was developed with Sujane and other people in the FDA. PCR, pathologic complete response, is a regulatory pathway identified most recently by the FDA. And I can't tell you about the signatures, but unless you ask. I think Kyle will say more about it and how we drop arms, etc. I do want to mention something about power in view of Andy's talk. Here, power is now much more complicated. There's no single power. So when we do simulations, we pretend that there's a drug that's beneficial in triple negative disease, say. That's one of the subsets. It's also that's a combination of subsets. That's one of the signatures. And we ask, can it pick out triple negative and say it's good in triple negative, but it's not good otherwise. That would be amazing. It's not so amazing for us because we're able to do that with reasonable power. But, so you could get the true signature. You could also identify hormone receptor negative disease, which is a bigger subset than triple negative, but includes triple negative. That's kind of a success. You could get only subsets that benefit, so you could get triple negative plus mammaprint high or something. That would be a subset. That's another kind of power. You could get at least one subset, and of course you could get none. So this is a generalization of the I-SPY 2. I-SPY 2 and 3 effects, match drugs with biomarker signatures, savings from common control, better therapies move through faster, drug biomarker pairs graduate to small focused, successful phase 3 using Bayesian predictive probabilities. And the offspring, I mean, it is incredible hype associated with this from other cancers, melanoma, colorectal cancer, but to Alzheimer's, HIV, acute heart failure, H1N1 pandemic. People are very interested in this kind of a thing. And software, there's FACS, there's SAS Adapt. This is a collaboration with very consultants, Tusela, who's in Cambridge and Abington, and SAS. And here's a solution design, and I gave you a whirlwind tour at the end, and I apologize for speeding up at the end. Thank you.
Okay, let's have one very quick question before we move to the next speaker.
So
Sure.
how do you see the involvement of adaptive trials within the industry? How do you see the involvement of adaptive trials within the industry? Is there more adaptive trials being sort of looked at from pharmaceutical companies, or how's that?
So are there more adaptive trials within the pharmaceutical company? So it's really, I mentioned the NET and the cooperative groups. They're really behind.
Yeah.
Oncology is ahead in some ways because of the biomarkers that they're looking at, but even they, in terms of adaptive design, are behind. So the government tends to be slower. The one exception, I think, is the FDA. The FDA has been hand in hand with the industry in developing the guidance and the ways to proceed, but it's been pushed by pharma, the pharma working group on adaptive designs. And there's great enthusiasm in much of the pharmaceutical industry, and they're actually, putting they're doing what they say they want to do, and they're developing adaptive designs. Most of them are in the early phases, but there are some that are in the later phases. Huge interest in biotech. One biotech person called me and said, I want to do a Bayesian design. And then he explained to me what he was about. He said, well, efficacy is clear. I'm going to see that after a few hundred patients, but I got to do safety. And the FDA tells me I need 800 patients for safety. And I said, well, you don't need an adaptive design. You just go to 800 patients and satisfy the FDA as to safety. And he said, well, you don't understand. My primary venture capitalist told me that he's not going to give me any money unless I do a Bayesian adaptive design.
Thanks.
Thanks.
Okay, let's think.